Thank you and good morning everyone, and I'm very honored to be here at the first session at the EAACI of the International Eczema Council, which we hope to make a good collaboration in the future as well. I joined as a counselor last year when we were in the American Academy meeting, and I was quite impressed by the level of science and the passion that they have for education and for networking with the other specialities. So I'm really grateful that they are opening to allergy specialty, and I hope that we will recruit more allergists in the future into this very nice network that they have. So my assignment for this morning is to set the scene, because if you are talking about safety of these newly introduced drugs, for us is not so much experience working with them as allergists, so we have to learn from our colleagues which have a little bit more experience. This is why we set the session focused on safety. So I would like to introduce you a little bit to the mechanisms and consequences if we target this particular pathway in the immune system. These are my disclosures. None of them are relevant for the talk that I'm going to give today. So I'm willing to discuss with you this morning where and how we can intervene in the Jak STAT pathway, a little bit about the relation of targeting this pathway and the concept that we call immune fitness. As you know, the immune system is very helpful with fighting infections, fighting cancers, fighting autoimmunity, fighting allergy as well. So we have to think of what the consequences of what we are doing. And then a little bit about the role of this pathway in atopic dermatitis, in asthma, which is new. It's not yet in current practice, but it will come. So I think that as allergists, we also have to understand where and how we can do this in asthma. And then, because this is my personal field of research, how to integrate with personalized endotype-driven decisions targeting this pathway. So, as you know, this pathway is a central communication node in the cell function. Why it is like that? It's because it's stimulated by a lot of substances. Besides, we are very focused on inflammation, and of course we think that and we all know that it's the common language for most of the interleukins. But there are a lot of other signals to this pathway coming from normal, components of our biology, like for example hormones or interferons, colony stimulating factors, and last but not least, biophysical forces influencing the cell. So that means that this pathway can have a lot of good functions, for example, involved in hematopoiesis, involved in the concept of immune fitness, that I will come back to that, in tissue development and repair, and in barrier function, which we know is very important in general starting the allergic diseases, chondroprotection, osteogenic differentiation, and many other more beneficial pathways that are there. On the other side, it promotes inflammation. We know it's a key pathway to promote inflammation, apoptosis, and on the metabolic side, adipogenesis, and it's a very important pathway, for example, driven through biophysical forces into cardiomyocytes hypertrophy. So a lot of research being done on this double-edged, role that this pathway is having in the immune system. Now, if we look at the specific structure of this pathway, there are 4 molecules that are important there, JAK1, 2, and 3, and tyrosine kinase 2. They all have a pretty similar structure with 2 domains. One is the kinase domain, where the JAK inhibitor is acting, which links the ATP and generates the reaction. It's held by the pseudokinase. And then 2 other important components of the molecule, which not only anchor it into the cell membrane, but ensure communication with the signal that comes from outside the cell. Jak inhibitors are expressed in a variety of cells and tissues. I'm not willing to go through this very nice scheme that EMA drafted recently, and I invite you all to look at this paper, which is really, really very informative. Just to let you know that for each of the cytokines, for example, you have a different pathway, and this is where selective inhibition based on the endotype that you would like to target could happen. So this is something that I would like you to keep in mind for our discussion further. Now, of course, being such a powerful node in cellular communication, there are always, mechanisms that counterbalance this activation, and some of them are here on this slide. So for example, starting from the molecular activation at the cell membrane, the CD45 is a very important molecule. Then we all know that for cytokine activation, the SOX is a very powerful inhibitor. Also for the JAK system itself, the PTPs are also important because they do dephosphorylation and they decouple after the initial activation the system. Now, how can we target this pathway? We can think of different levels of targeting, starting, for example, at the cytokine level that promotes inflammation, for example, using recombinant cytokines or blocking the cytokine or blocking the receptor where the cytokine is acting. Then comes the real target on this pathway with the use of JAK inhibitors acting at that kinase portion that I was mentioning. Or we could use, for example, the phosphorylation using peptide inhibitors or small molecule inhibitors, and then at the level of the transcription at the nuclear level, a lot of other interventions like decoy oligonucleotides, antisense oligonucleotides, and small inhibitory RNA. Well, that's the general overview of this. I'm sure there is more, but we are not at the immunology course here. So I would like to talk to you, because now we are slowly starting to talk about and think about the safety of this pathway. What about this pathway and the roles, or the physiological roles of the immune system, which are called the immune fitness? So let's look first at immune surveillance. Immune surveillance, which is, for example, extremely important against infections. And here type 1 interferons are one of the most important molecules. And the good news is that they do have a different pathway of activating this system, providing survival signals, antigen processing and presentation and proliferation, which are good things, together with antiviral response, immune regulation, antigen presentation as well, and checkpoint proteins. This is completely different from the other type 2 interferons, so the pathway is completely different through different signaling molecules which regulates the inflammatory response and the immune regulation. So we do have some possibility of selectivity here in terms of, guarding the good protective anti-infectious immune response and blocking the inflammatory pathways. The second part of the immune surveillance, of course, is looking at the tumors. And here again you have a very, let's say, good dichotomy between this system, with STAT5 being more and STAT3 being more involved into tumor growth and metastasis, and STAT3 being the ones that is helping the immune response against tumors. So again, we're doing MHC presentation and immune system activation, and of course it is harnessed now for speeding up the therapeutic response in anti-tumor immune modulation. So again, we do have a possibility of dissociating here and keeping the protection of the immune response that is through this pathway. Then something that is really recent and is really very nicely shown here, and again we have to have in mind that it's not only the effect on the infections or on tumors, but the immune system has a very important metabolic regulatory role, and we are all learning about immune metabolism, which is very, very important currently. So this is a very nice system showing that upon recognition of infection, this molecule, which is called UPD, is signaling to the cell, and the signaling happens to be through Jak STAT pathway, as we could expect, because there are a lot of metabolic molecules signaling through this pathway, as hormones do. What this system is doing is blocking and is inducing in the smooth muscle cell insulin resistance. That means that the insulin is shifted and the nutrients are shifted away from the muscles and driven to the immune system to fight infection. This is a very, very nice, mechanism that is happening, and we have to check the selectivity of these and what we are doing with our Jak STAT inhibition in the future. Now, I'm approaching the two diseases that we are currently treating as allergists, and the most knowledge that we have, because we already have the molecules in the therapeutic area, is the Jak STAT pathway in atopic dermatitis. And here there are a lot of important cytokines that we currently target for the treatment of atopic dermatitis, and I would like to share with you their signaling pathways and the actions that they do. So for example, IL-4, we know it's a very important T2 cytokine that is a signature cytokine for T2 profile. It signals through the path of JAK1 and then STAT3 and STAT6, and we know that it's very important because it induces IgE isotype switching, regulates T2 target Z genes, attenuates expression of important molecules in the skin barrier like filaggrin, lorigrin, involucrin, and claudin. Now, the second cytokine, which is pairing with IL-4, and they do a lot of things together, is IL-13. It signals again through JAK1, but it also uses JAK2 and tyrosine kinase 2, so it has 3 of the 4 molecules of the JAK system, and again STAT6 is there. It helps IL-4 in the IgE isotype switching, regulates special target genes for IL-13, is a major inducer of T2, which is independent of IL-4. This is extremely important.